early career folks, especially postdocs and senior grad students. And so we thought to ourselves, you know, young people don't always get a chance to have the opportunity to present their research. So we want to give them the focus, but at the same time, we're still going to allow some of the PIs to briefly discuss what their research is all about, give a nice overview. So we're going to start off today with Rory Black, who's affiliated as a PI with the PCE3 steering committee, and then we're going to hand it over to Zoe Todd and Zachary Cohen. But before that, I'd like to give my fellow seminar coordinators the chance to introduce themselves. So if you guys want to go ahead and jump in.
I'll start. My name's Becca Guth-Metzler, and I'm a graduate student at Georgia Tech, where I'm advised by Lauren Williams in the chemistry program and Jennifer Glass in the Earth and Atmospheric Sciences program. And my interests are with RNA and its relationship with metals. Since RNA has been preserving molecular history for billions of years, and it's also dependent on metals. So I'm investigating whether RNA can tell us about which metals were used on early Earth and how that usage has sort of shaped the face of evolution in life today. And with that, I'll pass it off to James.
Hi, I'm James Iguchi, and I'm in the NASA postdoctoral program, and I'm working with Timothy Lyons at the University of California, Riverside. And I'm interested in how, looking at how the cycling of carbon between Earth's surface and interior may have played a role in the atmospheric evolution, mainly of oxygen and the carbon cycle. And maybe our last organizer is Danielle.
I'm a postdoctoral researcher at Goddard in Greenbelt, Maryland, and I'm working in the astrobiology analytical lab studying prebiotic organic compounds in meteorite samples and other extraterrestrial materials. And with that, I'll pass it back to Al.
All right, thank you very much. And so that's enough from us. I'll hand it over now to Karen Rogers, our fearless leader, and she's going to tell us a little bit more about what PCE3 is really all about and put this seminar series in a bit more context. All right, I'll stop sharing my screen.
Thank you very much. Let me get my screen shared. Can we do this? Let me see. So I guess we should have tried this out beforehand. It's not letting me share my screen, Albert. I don't quite know what to do here. I can do the PCE3 introduction on the fly.
Let's see.
I'm not really sure why this is going on. The only options I get are one participant can share, multiple participants can share, and under advanced sharing options, who can share, only host.
Oh, James, can you make Karen a host, I think?
Yeah, I'll make her you were a co-host, but now you're a host.
Okay. Aha, okay, cool, awesome. Sorry about that momentary lapse there. So welcome everyone. My name is Karen Rogers. I'm one of the 4 co-leads of the Prebiotic Chemistry and Early Earth Environments Consortium. I'm so glad you're all here. I'm just gonna spend 5 minutes telling you a little bit about PCE3. And then I'm gonna turn it over so you can listen to these great talks by these 3 folks. So let me tell you first about what PCE3 is. We spent, I don't know, 6 or 8 or 12 months or something trying to come up with this sort of definition of who we are. But essentially, we're a group of scientists who work on origins research, everything from how stars evolve to how life finally emerges. And we're trying to get together so we talk better with each other, and we have more cross-disciplinary communication that will hopefully transform the origins research community in general. So that's what PCE3 is. It is not just our brainchild. This is actually one of 5 research coordination networks that was put together by the NASA astrobiology program. The first one to be founded was Nexus. That's the Nexus for Exoplanet System Science. I believe Nexus was founded in 2016. So it is sort of the original research coordination network in the astrobiology program. Since then, PCE3 was put together a few years ago. That's us. There is also NFOL, the Network for Life Detection, and now the Network for Ocean Worlds. There is one more RCN coming on sort of complexity of life and life's evolution. So hopefully you will be hearing from NASA Astrobiology soon to see what that 5th RCN is. But together you can see that largely what these 5 RCNs do is cover the NASA astrobiology goals in general, which are to understand the origin, evolution, and future of life in the universe, right? And so we think a really exciting piece of that group. And so we're glad to see you all here, because that means you all think that this is also an exciting piece of the astrobiology program. So who we are. Honestly, I wish I could sort of stop sharing my screen quickly and show all of your faces, because that's who we are. PCE3 is really just a group of people who are interested in this topic, who want to learn more and do more and get this community sort of invigorated and just talking more. So the community at large, that's all of you, and that's really the best part, I think, of PCE3. Not to put the rest of us to shame, but really we're here to hopefully just really facilitate the community at large doing more activities together and getting together more often. And so my co-leads are pictured here below: Ram Krishnamurthy, Tim Lyons, and Lauren Williams, and myself. The 4 of us are sort of the logistical go-between between you guys and NASA. So we essentially try and do that logistical sort of piece. And then we have the steering committee, and the steering committee is made up of PIs of NASA-funded projects that sort of have something to do with the topics within PCE3. And then there are the team members of all of those projects, right? And so they are sort of by definition part of the consortium, but really the consortium is made up of the folks and the scientists out there in the origins community who want to be a part of this endeavor to speak more clearly with each other and try and learn from each other, and really try and speak across some disciplinary boundaries that are sometimes really hard to charter, right? I mean, I came up as a geologist. I work on sort of prebiotic chemistry now, but we have people in PCE3 who do everything from stellar evolution to sort of phylogenetic backcasting, right? So looking at the phylogenetic tree and trying to understand what Earth's earliest life might have been like. That's a really wide breadth of disciplinary expertise to sort of understand. But we all kind of need to know everything in there, at least to some extent. And so our goal as a community is to try and facilitate bridging those gaps. So there it is, the science goal of this group is to investigate the delivery, synthesis, and fate of small molecules under the conditions of the early Earth and the subsequent formation of protobiological molecules and pathways that led to systems harboring the potential for life. So if you paid us per word there, it wouldn't be much, but if you paid us in time, that took us forever to come up with. Please don't tell us we missed something, because we'd be very sad. So that's the science goal, but the science goal is really facilitated by sort of our objectives. And this objective right here, I'm not going to read all these, they're on our website. The website is down in the right-hand corner. But the first objective is to integrate across the disciplines that are within PCE3 to help us speak to each other a little more easily, and collaborate to do new and innovative and sort of transformative research that this community hasn't seen before. So we can get to closer to an origins model that really does sort of match what we think the early Earth was like, right? And so we're striving toward what I like to call planetary plausibility, right? Has to be, if we're looking for the origin of life on this planet, actually has to sort of go along with what we see on this planet and what we do and don't know about what the early Earth was like. And so that objective I always like to put up and sort of have everyone look at, because that's why we're here. That's why we have things like workshops and seminar series, so we can integrate across these disciplines. Today is a great kickoff to that objective in general. So what is it that we do? A big part of our job is community and integration, right? And so we sort of leave it to everyone out there in the community to do their science. Our job is to help that science be more coordinated. And so there's community integration, sort of transdisciplinary collaboration, and some of that includes breaking down language barriers, uncertainties, and facilitating communication, and sort of expanding this cross-cutting research. So I'm not going to list all of our activities, but I wanted to just kind of give you a brief intro to some of the activities we've been doing more recently. The most recent one you're at right now, so welcome. You're now sort of at the very forefront of everything PCE3 is doing. Recently, back in October, November, we had our inaugural community workshop. It was called Building a New Foundation. We have another one coming soon on new directions, so I hope you can all join us there. We also contributed several white papers to the decadal survey. There is a few upcoming sessions at Goldschmidt, so that's first week of July, that you should all tune into. We'll list all of these on our website. We had submitted 3 sessions for AbSciCon 2021, which is now AbSciCon 2022. So you'll see those coming in the next year. And then this last thing is sort of anything you want it to be, right? The PCE3 community is what you make of it. We're here to facilitate. You're all here to do what you want with it. Anything you guys want to do, you are PCE3. We're here to help you make that happen. But what happens doesn't come from us. It really does come from all of you. Whoops, one more thing. So on that note, I sort of want to put out a call for anyone who wants to sort of come play with us. We have lots of things that we think can be done, and there's a bunch of stuff I'm sure we haven't even come up with yet. So if you're interested in what folks have done in the past, we have the seminar series organizing committee who just introduced themselves. Thank you, everyone. Our inaugural workshop organizing committee that can be found on our website. You can ask them sort of what that was like. And I put that out there because there's a second workshop coming out, and we have a call for folks to come help with organizing that second workshop. We're also putting together an outreach and early career group. So if you can, if you'd like to play with the outreach and early career group, we'd love to have you. And then, you know, this last piece at the bottom is any other activity you think PCE3 should be doing, because this is your community and your consortium. So reach out to us, and if we can make it happen and you want to sort of put the blood, sweat, and tears in, we're happy to help you make that happen and make PCE3 a really vibrant community. So that's my intro to us. I want to have a big shout out and thank you to the seminar series organizing committee. You guys are phenomenal. Really so excited to see what's coming, and I hope to see the rest of you every week. So that is it. If I can figure out how to stop sharing, that would be great. There it is.
All right. Thank you, Karen, for that fantastic introduction and giving us some context about where PCE3 has been and where it's going. All right. So now it's my great pleasure to introduce our first speaker, Roy Black, who is a PI, part of the PCE3 steering committee. So he's going to give us a brief overview of some of his research. And so over a 26-year career in biotech industry, Roy built and led R&D programs that identified important enzymes associated with inflammation. And in 2010, Roy took a sabbatical to learn about prebiotic membranes in the laboratory of Dr. Dave Deemer, who I'm sure a lot of us already know, at the University of California, Santa Cruz. And in 2011, he left the industry to pursue his original life interests as an affiliate professor at the University of Washington, where he studies model prebiotic membranes, which I think he's probably going to tell us about. So Roy, if you'd like to share your screen. All right.
Okay. Trying to make this full screen. There you go. Okay.
Perfect.
Thanks to James and Albert for the opportunity to lead off this seminar series. Today we have an all University of Washington cast. And a major focus here is the role of prebiotic membranes in the origin of cells. So I'm going to start with a brief overview of this effort before turning it over to Zoe and Zach. Our central hypothesis is that cells, which are this really odd combination of the two biological polymers and a membrane, arose from a self-assembled aggregate of the building blocks of these three components. And of course, the building blocks came from the prebiotic soup. We find this scenario more plausible than the conventional view that cells arose from preformed polymers and membranes. We see a number of challenges for this view. First, how could these components have formed separately? And of course, there are many answers out there to this question, but I think it's fair to say none of them have been generally accepted yet. And second, even if these components could form independently, how would a replicating system of RNAs or RNAs and proteins then get into a membrane compartment and not only get in, but continue to function there as before? In contrast, a self-assembled, an aggregate of the building blocks readily explains how the two polymers wound up associated with membranes. These three components co-localized because their building blocks had first self-assembled. And this scheme helps to answer several other long-standing problems in the field. So over the past 10 years, we have established evidence that such an aggregate does form, and we've shown that it is more stable than bare membranes. We're currently focused on this arm of the scheme. We're testing whether such an aggregate does actually facilitate formation of the biopolymers. And we're testing whether a oligomerization of building blocks further increases stability of the aggregate, thereby driving the progression toward protocells. So that's where we're at, and Zach will explicitly talk about some of that work, and I think Zoe will also touch on it. So I will turn it over to them.
All right. Thank you very much, Roy. That's definitely some exciting stuff. And for those of you in the audience, we'll do questions at the very end. So if you have a question for Roy, after the last talk, we will open up the chat and you guys can pop in some questions for any of our speakers. All right. Thank you very much. So now it's time to hand the floor over to the stars of our show, the early career researchers. So first up, we have Zoe Todd. So Zoe Todd is a first-year NASA Hubble postdoc fellow, specifically a Sagan fellow, at the University of Washington. And she completed her PhD in astronomy at Harvard University in the lab of Dimitar Saslov. And she also spent a lot of time in the Shostak lab as well, where I had the great pleasure of being able to work with her. And she got her undergrad education at Penn State with majors in astrophysics, biochemistry, and physics. Zoe's research interests have been geared towards astrobiology and in particular the origins of life. So Zoe uses a combination of chemistry and planetary science, as well as both experiments and theory, to provide an improved understanding of the circumstances that could lead to the origins of life and the implications for life on other planets. So Zoe, if you'd like to start sharing your slides, the floor is yours.
Wonderful. All right. Hopefully, can you see that? Is it full screen?
Yeah, perfect.
Awesome. Thank you. All right. Well, I'll start off by saying thank you so much to the organizers for having me here, and thank you all to those of you attending, and thanks Roy for that great introduction and really fascinating overview. So as Albert said, and thank you for the kind introduction, Albert, that was wonderful. I'm a Sagan Fellow at the University of Washington, and I'm interested in precisely, I think, what the PCE3 community is also interested in. So today I'll be telling you a little bit about what can the planetary environment tell us about the origins of life. So if you look at the scheme that I've sketched out here, everything in my talk, I'm not claiming that it is the way that life originated on Earth, but I'm claiming that perhaps it is a possibility. So I'm interested in understanding how we can go from very simple feedstock molecules that are readily available on the environment in either the early Earth or other exoplanets if we start thinking about the search for other life. So how these simple geochemically abundant or available feedstock molecules can come together and assemble in prebiotic chemistry to form the precursors of life, so the biomolecules that we would need, and then further how those biomolecules would come together, polymerize, and assemble, be able to self-replicate, and be able to be encapsulated in some sort of protocell to go on to form what we might think of as the first type of life. So with that in mind, I'm going to sketch out examples of each of these steps here as stuff that I have worked on in the past or am working on right now. But again, there are so many theories out there, so many thoughts, and I think as a community coming together to investigate these ideas from all sides, we stand to make a lot of progress and hopefully understand more about our own origins and what this means for the possibility of life on other planets. So it's a very exciting time to be an astrobiologist and to be interested in origins. So I'll start out by showing this cyanosulfidic photoredox network that comes out of the lab of John Sutherland. So this scheme right here is from the Patel et al. 2015 paper. And I'm sure there are many people in the audience who are not chemists like myself. So throwing up this many chemical structures could be terrifying to them. I'm sure there are also many chemists in the audience who think that this is very simple, and I will endeavor to not frustrate you as we go through this next step. So I'm going to walk you through this network. But the really nice thing about this cyanosulfidic photoredox network is it can synthesize all four major types of biomolecules used by life today. So you can get sugars, amino acids, lipid precursors, and ribonucleotides, all from one sort of uniform set of circumstances. So let's step through that a little bit. All right, so in this pathway, you can make 2 and 3 carbon sugars, glycolaldehyde and glyceraldehyde, and these are molecules on the way towards making many of the other further, more complex molecules in this network. You can also make lipid precursors, so on the way towards hopefully synthesizing some sort of phospholipid or something that can make a membrane to become a protocell, which we'll hear a lot more about from Zach's talk, I believe. Then we can also, in this network, they also make amino acids. There are 12 total here, I believe, if I can count correctly. So this actually does better than the Miller-Urey experiments that can synthesize amino acids from spark discharge. Finally, you can get pyrimidine ribonucleotides. So these are 2 of the letters of RNA, C and U. This scheme does not have an explicit synthesis of the purine ribonucleotides at this point, but there's been a lot of recent progress on there, and hopefully I have faith in the chemists to figure out these very challenging problems. So you can make these 4 different types of biomolecules, but where do they come from? So we can look to the name of this network. It's a cyanosulfidic photoredox network. So let's break that down a little bit. The cyano refers to hydrogen cyanide, which I've boxed here in red as it comes into the scheme. So you'll notice that HCN is the starting point of this network, essentially. So almost every single carbon and nitrogen that you see is derived from HCN ultimately. Then if we think about the second part of the name, cyanosulfidic. So sulfidic gives us a clue that sulfur is involved. So here in this scheme, they use H2S, sulfide. Some recent work has moved to sulfite, which is SO32- instead. But so a source of sulfur in combination with the cyanide makes this cyanosulfidic network. Now, finally, the last part is the photoredox part. So photoredox refers to the fact that this network uses UV light in a few key places, either to drive the synthesis, drive the network forward, or to perform some key steps, including down here, the conversion of C into U, and do some selective degradation as well. So that was your whirlwind tour of the cyanosulfidic photoredox network. So I will focus on this type of prebiotic chemical network with the note that this is not the only way that origins of life could have occurred. There are many other thoughts out there, could be equally as valid. So, but this will be the focus of some of the work that I'm talking about today. All right, so now we have that cyanosulfidic network and looking at this picture here. So if we want to understand how could that cyanosulfidic network that has been elucidated through chemistry in the laboratory, we want to understand, is that plausible in early Earth environments? Could it be possible on other planets? What can the environment tell us about the chemistry, and what can the chemistry tell us about the environment? So I've kind of broken down a very simple sketch from feedstock molecules into precursors and finally into protocells. So if we focus on the first step, where do feedstock molecules come from in a planetary environment? There are many possibilities. There have been many suggestions throughout many years in the literature. So you can do atmospheric photochemistry, you can do lightning generation, you can do impact synthesis and delivery, or a variety of other geochemical sources. So the one example that I'm going to be talking about that I've worked on in the past is going to be impact delivery. So we just saw from the cyanosulfidic network that is going to be central to my talk today, that hydrogen cyanide as a source molecule is quite important. But where does that hydrogen cyanide come from? Many of the sources that I just mentioned, atmospheric photochemistry, lightning, or impact synthesis, can be used to synthesize HCN in an early Earth atmosphere, probably. But we turned to a potentially different source of HCN that does not depend on the redox state of the atmosphere. So if you take these big telescopes like ALMA in Chile and South America, turn them to the night sky, you can see HCN in protoplanetary disks. You can detect HCN in comets. And we know that the early Earth and the inner solar system got hit by lots of material. All you have to do is look at the moon and see all of the craters on the moon, see craters on all of the other planets in the inner solar system as well. So the question then becomes, if you have HCN in space, you have HCN in comets, you have some comets potentially hitting the early Earth, could any of this HCN survive impact and be delivered? If it does survive, what concentrations could it lead to and what timescales is it around for? And then finally, is this relevant for prebiotic chemistry? So this was the question that we sought to answer. This is something that Karin Oberg and I worked on together. I give her a lot of credit for that. So the model that we invoke here, we assume a spherical comet broken down into 6 different radius shells and then model the different temperature profiles that this comet undergoes, taken from past literature simulations. We give our comet 4 molecules initially: water, HCN, CO2, and CO. So the first thing that will happen is as the temperature increases, you can thermally decompose these 4 molecules into their reactive radical components. So then those of you who are chemists will know that radicals react very readily. Lots of chemistry can occur from radical chemistry. So there are a lot of reactions that can then happen. In particular, reactions of HCN with either OH radical or O radical oftentimes lead to the destruction of HCN and of that nitrile bond that is quite important. Sometimes, though, they don't initially destroy the nitrile bond. Like here, you still have CN with the CN triple bond, but there can be then further nitrile chemistry that can either return HCN or ultimately degrade it. So you might think 4 molecules, this must be really simple, right? Well, when you include all of the radical reactions to as much completion as possible, and we do believe that we were complete with respect to HCN as much as possible, you get something like, I think, 80 or so reactions encoded in this model. So another thing to consider here is that the impact angle, the velocity, and the radius of the impactor also matter. So a more oblique impactor that kind of skids along the surface of a planet rather than coming in straight, dropping down at a 90-degree angle, at that more oblique impactor and at lower velocities and smaller radius impactors, those will experience lower overall temperatures, which will lead you to increased survival of the molecules that it initially has, including that HCN that we were interested in. So when you take a fiducial impactor with a radius of 1 kilometer and a velocity of 20 kilometers per second, and then you integrate over the probability distribution of impact angles, and further again integrate over the volume of these different radius shells, we find that you can get about 2.3% of the initial hydrogen cyanide survives for that impactor. So you might then be asking, 2.3%, is that a lot? Is that a little? Is this relevant? What does that mean? And I would say that is an excellent question. So in the global context of HCN, the other unknown in here is that we don't know exactly how many comets or how many bodies that would have contained HCN hit the early Earth. This is quite unconstrained. You can try to estimate this based on isotopic constraints like the D to H ratio in comets and in oceans and match something from there. You can try to model it. It's pretty wide open assumption at this point. So limits have been suggested between like 0.1 and 10% of Earth's ocean water being delivered in comets. So if we take 1%, kind of a middle of the road estimate there, you can get something like 10 to the 6 kilograms of HCN delivered per year if you're assuming a 100 mega-year bombardment period. So what does this mean? Is this relevant at all? Well, to answer that question, we can then compare to the other potential sources of hydrogen cyanide. So from atmospheric photochemistry, this is dependent on the redox state of the atmosphere. So in a methane-rich or highly reducing atmosphere, you get 10 to the 10 kilograms per year. So this is orders of magnitude higher than the estimate that we arrive at for impact delivery. In a low methane or more oxidizing atmosphere, this drops by a few orders of magnitude. For lightning generation as a source, again, this is redox dependent. So more reducing, you get higher yields. 10 to the 9 kilograms in a mildly reducing CO-dominated atmosphere, you're about the same order of magnitude as what we found for impact delivery. And in an oxidizing atmosphere, it's lower here. So takeaway from this, impact delivery is not the most abundant source of HCN that could be possible on a planet, but it is a source of HCN that is perhaps not irrelevant, that is also not dependent on the redox state of the atmosphere. And then even further, if we don't consider the global scenario of impact delivery of HCN, but instead look at a local impact delivery of HCN. So here we're considering the case of if you have a piece of land and a comet comes in and impacts that land, makes a crater, and dissolves all of its delivered surviving HCN in all of the water that it delivers. So this is an upper limit to what we think the HCN concentrations could be. So in this case, if you go to lower impact angles, so 5 degrees, 15 degrees, or 25 degrees, the initial concentration of HCN are shown by these solid lines here. Then HCN will degrade over time. It will hydrolyze first to formamide and then to formic acid. The rate of this hydrolysis is dependent on the pH and the temperature. So the arrows here are showing you the amount of HCN concentration after 100,000 years. So at pH 7, the hydrolysis is fairly rapid, so you don't really have much HCN retained after 100,000 years. At pH 6, you can have some, and at pH 4, the lifetime is fairly long. So boiling this all down from this complicated plot to the main takeaway point here, if you want HCN in greater than millimolar concentrations, you can have that for about 200 to 300,000 years for the most favorable local impacts. You can have HCN above micromolar concentrations for 500 to 1,000 kiloyears. So I also have a prebiotic chemistry hat where I am in the lab pipetting around and playing with hydrogen cyanide, which is good fun. And I always say, if I can get my experiments to, if on the early Earth, you could give me millimolar concentrations of HCN for hundreds of thousands of years, that sounds pretty darn good to me. But I'll leave the debate of that to actual chemists if they desire. So takeaway from this, impact delivery of HCN could be relevant for local scenarios, especially under favorable conditions. Maybe it's relevant on a global scale, but there are other sources of HCN as well. So that was one foray into this initial part of looking at where do the feedstock molecules for prebiotic chemistry come from. So then moving on to the next step in this simple scheme, how do these feedstock molecules react together in the planetary environment to form building blocks of life? So I already mentioned that cyanosulfidic network, and that network and those chemists are phenomenal for getting to the biomolecules of life, and I give them all the credit for elucidating that wonderful work. But then if we look at what sort of constraints or what sort of plausibility can we put on this network, that's something that I'm interested in, and I'm going to share some upcoming and current work of mine. So we already saw that in the cyanosulfidic network, you could take HCN, you can make things like amino acids, sugars, ribonucleotides, and lipids. Now, breaking this down a little bit more into the mechanism of how this works through something called reductive homologation, where the production of solvated electrons through UV irradiation of reagents like ferrocyanide, this FeCN6 4-, and sulfite, SO32-, can drive this chemistry. So you have UV irradiation of these reagents, gives you solvated electrons, which react with HCN to form these biomolecules of life. And that's how that cyanosulfidic network is mainly driven. So then if we want to put this into the planetary context and assess some constraints on the environment, we can think about, all right, well, now you need this ferrocyanide and you want it to be doing this reaction here to produce these solvated electrons, which is driven by UV light. So where does ferrocyanide come from? Well, if you have a source of HCN, which is also needed for the cyanosulfidic network, and as I just shared with you, could be delivered through impacts or from other various planetary processes. And if you have ferrous iron around, as you probably did in large amounts on the early Earth before the oxidation of the atmosphere, these can react together to form ferrocyanide. There's also another reaction that occurs with ferrocyanide and longer wavelength, UV light and visible light, to generate what is called photoacquated ferrocyanide, where basically you're taking off one cyanide and putting a water in its place. And over time, this degrades slowly to be lost from solution. So someone like me would look at something like this and ask questions like, well, how fast does ferrocyanide form under various conditions? How stable is it? Because we want this branch of the pathway to be occurring to do this productive chemistry. So how fast is this? Then also, how quickly is ferrocyanide lost due to this non-productive long wave radiation through this pathway here? Because effectively, this is all happening at the same time and in equilibrium. So if we want to understand how to let this occur, which is what we want to be happening on the early Earth for prebiotic chemistry, we need to understand both the source and the sinks of this molecule here. And this is by no means complete with respect to ferrocyanide chemistry, but this is what I'm going to be sharing with you today. So what we did is we looked at the UV degradation of ferrocyanide due to this long wave radiation. So these are, again, the two different channels, the photooxidation, which is productive, what we want to have happening, and then the photoequation, this destructive loss of ferrocyanide. So this here is the absorption spectrum of ferrocyanide, and under continuous irradiation with UV light, you can see it's been irradiated for 60 minutes here, the absorption spectrum decreases. We can use this absorption spectrum to calculate the concentration of ferrocyanide as a function of this irradiation time. You get this nice, beautiful straight line, and you get the rate constant of the reaction is going to be the negative slope of this line here. And I want to definitely give a shout out to Corinna Kufner and Gabby Lozano, who helped with these experiments and were instrumental for doing this part of this project. So the other thing to consider is that this UV-driven degradation of ferrocyanide likely depends on the wavelength of the UV light. So what Gabby and Corinna did very wonderfully was looked at the irradiation wavelength dependence of this reaction. So they did the reaction between 290 and 390 nanometers and we determined the rate constant as a function of this irradiation wavelength here. So you can see that the reaction is faster. You get more destruction of ferrocyanide at shorter wavelengths and less at longer wavelengths. So now we can ask, what does this mean in the UV environment on the early Earth? So we can use models of the surface intensity of solar light reaching the surface of the early Earth through a sample N2 CO2 atmosphere. This was taken from Ranjan and Saslov 2017. And then basically integrate up this yellow line and in the same 10 nanometer bins as the experiments were done, multiply by the points here, And now you're getting these points shown in this right-hand panel. So this is showing you that since there's more light coming at these longer wavelengths, the relative importance of those as the degradation of ferrocyanide increases, but it's still not greater than these shorter wavelength photons that are primarily doing the destruction under the early Earth UV environment. The other cool thing that we can do with this is then integrate up these points here to get a relative rate of the reaction given the UV surface intensity on the early Earth that we expect. And when you do this, you get something on the order of minutes for these conditions here. Now, the other caveat is that these reaction rates are going to depend on the pH and on the temperature and on the concentration of ferrocyanide in the solution. So we also determined the dependencies of the rate of destruction on those three factors, pH, temperature, and concentration. And then when you put this all together, combine it all, write up a simple model, we can get plots like this, where here on the left, I'm showing you for 1 millimolar ferrocyanide, the lifetimes as a function of temperature on the x-axis and pH on the y-axis. And on the right, we're showing the same thing, but for 5 millimolar ferrocyanide. So to give you an idea of what this scale bar is, so your darkest blue is something like 2 hours, 120 minutes. And your lightest green or yellow, whatever color you want to call that, is on the order of 5 or so minutes. So we're not talking about this being stable for days or months. We're not talking about it degrading in the matter of seconds. It's somewhere on the order of minutes to hours. And so again, we can see what conditions favor the retention of ferrocyanide due to this long wave radiation. And we find that that's going to be basic pHs and lower temperatures, as well as higher concentrations. So now this becomes a question for how fast does your productive chemistry occur? Does it take hours and days, or does it take seconds? And in which case you can evaluate what sort of conditions would allow you to have that ferrocyanide in UV irradiated environments on the early Earth. So that's an element of how we can take that prebiotic cyanosulfidic network and then try to put it into a planetary context and understand what the environment is constraining about the chemistry, and further what the chemistry can constrain about the environment. So going back to this picture here, this was an example of how we can kind of examine this second step in the pathway, how you go from the feedstock molecules towards building up the precursors and the biomolecules of life. So moving on one step forward, it seems like it takes a lot of work to get to the synthesis of those biomolecules. It definitely does, but even once you do, you're not done. We still don't have something that is life. We still don't have, life on Earth. So to get there, what you would have to do then still, or what still remains, is polymerization of these biomolecules into the polymers like RNA, peptides, whatever else you need. And then you also need to get those to replicate, probably chemically without enzymes that life uses today. And then assemble them inside of perhaps a membrane or some sort of component that can encapsulate them in order to allow Darwinian evolution to eventually come about and occur. So it's this transition to protocells that I'm calling this third step here, and there's a lot that goes into that. So I'm not going to describe too many details about work that I've specifically done on this step, but I'm going to show you work that I'm thinking about and in the process of doing, and don't have too many results yet, but it's some interesting questions that we would hope to answer. So as Roy had mentioned in his slides, so if we have these fatty acid vesicles that spontaneously self-assemble in water, there's a great advantage to thinking that these could be your first initial protocells. They can grow when you add micelles or when you add excess fatty acids to them. They can divide if you agitate them. There's been a lot of work on that. But so Roy has been part of a group that has shown that you can actually get various molecules binding to these membranes and binding to these vesicles. So now the question becomes, if you have an empty vesicle versus something with bound peptides or bound molecules, whatever you want, is there a difference in the growth rates of these two vesicles here? And if so, could that lead to an evolutionary advantage? And could you perhaps get some other interesting, like, can you synthesize polymers by having them anchored to the membrane or within the membrane? So we're currently examining that and looking into that. Hopefully we'll have some results soon. Then another question that I've been interested in from a long time is, can protocells protect encapsulated contents from UV light? So I've described some work that shows how UV light is necessary or is good for driving some of this prebiotic chemistry. It's the source of energy for that cyanosulfidic network. However, we also saw that UV light can degrade ferrocyanide. That's a potential constraint. We're also probably more familiar with the damaging effects of UV light. If you go outside on a sunny day, you get sunburned, that is effectively UV light damaging your biomolecules, damaging your DNA. So you reach this kind of catch-22 where maybe in this scenario, you need the UV light to do the productive prebiotic chemistry, but also the UV light can damage the products that you make. So is there a way to resolve this? Perhaps it would be really interesting if by having perhaps your RNA nucleotides or polymers encapsulated inside of fatty acid vesicles, you could shield it from the damaging effects of UV light somehow. So the mechanism for thinking this could potentially be worth investigating is that if you have a photon coming in, there are essentially three things that could happen to that photon. It could be scattered away just due to the size of the vesicle and the wavelength of the photon itself. It could be absorbed by either the fatty acid membrane or potentially something else localized inside the membrane, or it could be transmitted to the inside and perhaps then still do either productive UV photochemistry or destructive damaging UV photochemistry. So understanding this balance and understanding if there's a difference between encapsulated versus unencapsulated and the reaction to UV light is also something that we're working on and trying to figure out. So going back to this image that you're probably all sick of because I've showed it numerous times by now, this is essentially summarizing my talk here. So we've been looking into what can the planetary environment tell us about origins of life, hoping to constrain a fairly continuous path from feedstock molecules to precursors, biomolecules, and eventually to the formation of a protocell that can be self-replicating and start undergoing Darwinian evolution. So this is by no means the only way to get to life. I welcome other theories and think that everyone's take on this is definitely valuable and can teach us a lot. And we have so much left to learn, but this is my take on where we're going and what I hope to be able to do. Many people to thank for this work. Karin Oberg for her help with the first project, Impact Delivery. David Catling, he's my host at UW, wonderful help for the second part of the project. And Dimitar Sasalov, my PhD advisor, Jack Shostak, who let me in his lab during my PhD. I especially want to thank Sarah Keller and Roy Black for welcoming me into their lab at UW and allowing me to do some of these lipid vesicle work. Zach as well, who you're going to hear from next. I'm super excited for his help with vesicles, and then Corinna and Gabby, who helped with the radiation wavelength dependent experiments, funding agencies, NHFP, Simons, UW, and the CFA. And of course, as always, my pony Peter makes my acknowledgements. This was him on his 10th birthday, I believe, a couple years ago. So that's the end of my talk. I'll be happy to take any questions at the end, but for now, I guess I will turn it back to Albert and then be ready to listen to Zach.
All right, thank you very much, Zoe, for that fantastic talk. I really love the picture of the pony at the end.
Thank you.
And yeah, like Zoe said, if you have any questions for Zoe, please make a mental note or write them down. After Zach's talk, we're going to open the floor to questions. So thanks again, Zoe. And now for our second star of the show today, we have Zachary Cohen. It goes by Zach. So Zach Cohen is a fourth-year PhD student in the chemistry department and astrobiology program at the University of Washington. And before coming to UW, Zach completed his undergrad in biology and physics at University of Illinois, Urbana-Champaign. As an undergrad, Zach used computational tools to research microbial communities and microbial genomes. During his time at University of Washington, Zach has been investigating potential roles for fatty acid membranes during the origin of life on Earth using a variety of experimental techniques, including mass spec and microscopy. And it looks like Zach has already shared his screen, so please go ahead and take it away.
Okay, thanks, Albert. Yeah, so and thank you to the organizers for giving me this opportunity. I'm really excited to tell you about all this research that I've done on fatty acid membranes and how they encapsulate solutes and allow peptide formation in early Earth evaporative environments. So the goal of my research is to investigate whether the origin of life could have occurred within evaporative environments on the early Earth. So to start this talk, I'm going to introduce the idea of a protocell as this early stage in the origin of life on Earth. And then I'm going to explain how I've been thinking about and modeling early Earth evaporative environments. And the rest of my talk is going to focus on 3 specific research questions. So the first question is, do membrane compartments function in salty conditions? Second question, do membrane compartments function after complete dehydration? And third question, are membrane compartments compatible with biopolymer formation during dehydration? And I'll unpack those all more. So fatty acid vesicles are plausible protocell compartments. This molecule on the left here is, hopefully you guys can see my cursor as well. So this is decanoic acid. It has a 10 carbon hydrophobic tail and an acid head group. And so we usually draw it as this ball and stick model. So decanoic acid itself is the prebiotically plausible compound. It has been delivered to Earth by meteorites, and people are actively investigating pathways to synthesize it abiotically. One way that has been shown to work is called Fischer-Tropsch synthesis. So because of the hydrophobic and hydrophilic regions in this molecule, it can assemble into membranes. So this is a picture of a vesicle, which is this like connected, in this case, it's supposed to be a spherical membrane compartment. And here's what some of these vesicles look like with the fluorescence microscope picture. And so just in case it's not clear, I'll highlight all those vesicles here, and I'll show you many more pictures like this. So these fatty acid vesicles are important because they're hypothesized to be compartments for the first protocells, which again are this hypothetical early stage in the origin of life on Earth, where fatty acid membrane is a compartment for associated RNA and peptide oligomers, and all of these structures would interact in important functional ways to give you cell-like chemistry. Now, it's important not to confuse fatty acids for phospholipids, which make up the membranes in modern cells. So you can see here in this box, this phospholipid is structurally, it's a very different molecule than fatty acids. And I'll talk about some ways that the behavior of these 2 kinds of membranes is different. So evaporative environments are great places to think about the origin of life because they can support very diverse prebiotic chemistry. So when I'm imagining this, I'm imagining a lake on land on the surface of early Earth, and there are solutes like fatty acids and like various salts dissolved in this lake water. And as the water evaporates, those solutes become more and more concentrated until they reach their solubility limit. And so then you're left with a saturated solution. When it comes to the salts, we're in my kind of model systems, we're specifically thinking about sodium bicarbonate and sodium chloride because we think they'd be ubiquitous in these environments. Sodium bicarbonate you can think about as being sourced from CO2 in the atmosphere. So then further evaporation would give you a completely dry lake bed, and then we imagine it floods and you go through cycles of wet and dry, wet and dry, wet and dry. So a couple key advantages of thinking about prebiotic chemistry in these environments is that they're accessible to photochemistry. Second advantage, cyclic dehydration and rehydration has been shown to promote condensation reactions in many experiments. And I'll talk about some work that I've done on that topic. And then number 3 is that evaporation within these saturated sodium bicarbonate lakes is suggested to concentrate phosphate and ferrocyanide salts, which are these very reactive precursors, and they're important in many of these model prebiotic syntheses that Zoe was just talking about. Okay, so that's just some background to set the stage. My goal here is to test whether protocells are a viable model within early Earth evaporative environments. So I'm going to focus first on this question that I've highlighted: do membrane compartments function in salty conditions? So we know that salts can cause fatty acid vesicles to flocculate. I'm guessing that's a term most people are not familiar with, so I'm gonna try and unpack that. Let's think about our standard fatty acid vesicle population. I've drawn that right here with vesicles of different sizes and different number of layers. In all cases, the vesicles assemble when the pH of the surrounding solution is near the effective pKa of fatty acids within the bilayer. So what does that mean? That just means that within the membrane itself, if we zoom in, the membrane is composed of protonated fatty acids and deprotonated fatty acids. So the deprotonated fatty acid is going to be negatively charged, and the protonated fatty acid is going to be uncharged. And as a result, each vesicle or vesicle layer here has a net negative charge on it. And that helps stabilize the vesicles. By charge repulsion, it keeps them apart. When you add salt, specifically monovalent cations, the effect of that can actually be to screen charges on the membrane. And so this will effectively make each vesicle uncharged, and then the vesicles can aggregate with each other just due to the hydrophobic effect. And so that is the phenomenon of flocculation, this aggregation due to charge screening. So I'm going to refine this research question a little bit and ask, can flocculation be prevented in saturated salt solution? And what I've seen in my experiments is that flocculation seems to be inevitable in saturated salt. So here I've got 2 different concentrations of sodium bicarbonate, a relatively dilute solution, and then a saturated concentration. This is about 1.5 molar sodium bicarbonate. And I was interested in if we could determine if there was a fatty acid to cation ratio where, below which we would not observe flocculation and above which we would. So I've got 2 different fatty acid concentrations. And also, to make a note, in these experiments, I've included decanol and glycerol monodecanoate, which are 2 additional prebiotic amphiphiles. So anyway, low and high fatty acid concentration. In the low fatty acid concentration and low salt concentration, I see a few vesicles. That's these circles in the image. And as I increase the fatty acid concentration, I see many more vesicles, which is consistent with what you'd expect. Keep the salt constant. But when I increase the salt concentration to be a saturated sodium bicarbonate solution, then it doesn't seem to matter what the fatty acid concentration is. In both cases, I just see these clumps. And so each clump here is just an aggregate of a bunch of vesicles stuck together. So regardless of the fatty acid concentration, in the presence of saturated sodium bicarbonate, I've just seen flocculation. Now, previous work from our lab that Roy and Zoe both alluded to has shown that small molecules like amino acids can actually bind to the surface of these vesicles and make the vesicles themselves more resistant to salt-induced flocculation. And that's true, but that's true at intermediate salt concentrations. So at saturated sodium bicarbonate, flocculation seems to be inevitable. So that motivated the next question, which is flocculation problematic for protocells or for the origin of life? And the answer seems to be not so much. What I've seen is that flocculation does not disrupt encapsulation by these vesicles. So here I've prepared vesicles with the fluorescent dye in the aqueous solution. So inside the vesicle itself, I've put a fluorescent dye. And then before I image these vesicles, I'll put in a hydrophobic dye to stain the vesicle membrane. Now, you might look at this picture here and say, why is it this filled circle? I was expecting it to be hollow. And the reason is just that a lot of times we're looking at these multi-layered vesicles. And so that's why this image appears to be shaded in the vesicle dye channel. So then I'll add salt to these vesicle solutions. So in this case, I've added enough salt so that it's the final sodium bicarbonate concentration is saturated. So this is a solution saturated with sodium bicarbonate. And in this case, it's a solution saturated with sodium chloride. In both cases now, I just see these clumps. And if you look at the scale bars here, these images are a lot more zoomed out. So each of these clumps is big, and it's basically just an aggregate of many, many vesicles. So this is flocculation. What's important in this middle panel of images is that the vesicles are still bright in the aqueous dye channel. So all of these flocculated vesicles, the fluorescent dye that was initially inside of them is still associated with the flocculated vesicles. When I dilute the salt, the aggregates break apart, individual vesicles are stable again, and I see that some of those vesicles still have a high concentration of the aqueous dye inside of them. So the encapsulation by the vesicles is maintained throughout the flocculation and dilution process. Now, I wanted to be a little bit more quantitative about this. So I tried to measure the rate of leakage. And what I found is that the leakage through the membrane actually does not increase due to flocculation. So I'm just going to explain how I did these experiments. I prepared a population of vesicles with a very high concentration of fluorescent dye inside of them. And so the concentration is high enough so that this dye is self-quenched. So the fluorescence is very low from the dye molecules inside the vesicle. As time goes on, some of those dye molecules can leak through the membrane and get outside and become more fluorescent in the outer solution. And I can measure that. The other important thing I can do is to add a chemical called Triton X. And Triton X is going to disrupt the vesicle membrane and induce immediate release of all of the encapsulated dye. So if we look at this first panel on the left, I'm plotting fluorescence intensity on the y-axis as a function of time. And if from the shaded squares, so in this case, I have not added any additional salt. So this is just nice vesicles, and I measure an increase in the fluorescence as dye molecules leak out through those vesicles. That's what I'm showing in the shaded squares here. So the increase in the fluorescence is very slow. It's a very small increase in fluorescence at first, which corresponds to a small rate of leakage. But at its final time point, I add Triton X and induce complete release of all the dye, and I see a large increase in the fluorescence signal. And this actually matches up with these unfilled squares, which is where I took a parallel aliquot of vesicles and added the Triton X right at the beginning. So the upshot from this kind of data is just that the time until complete leakage is a lot longer than the length of the experiment that I can do. So leakage is certainly, slower than the timescale of this experiment. And that's true for vesicles without additional salt. It's also true for vesicles in the presence of saturated sodium bicarbonate and saturated sodium chloride. In both cases, in these flocculated vesicles, the rate of change of the fluorescence is very small until I add the Triton X and induce complete leakage. So the time until complete leakage is a lot longer than the length of the experiment. So, okay, again, the upshot here is flocculation is not really problematic because encapsulation by the vesicles is maintained. Now, you might wonder, what about phospholipid vesicles? What about the membranes in modern cells? So here I've prepared a sample of phospholipid vesicles and added enough sodium bicarbonate so that it's a saturated solution. And I see that these vesicles still flocculate. Phospholipid vesicles flocculate as well. That's what we're looking at in this micrograph. So that gives us a satisfying, we know that vesicles can form in relatively dilute solutions, but this gives us a satisfying answer to the question of whether vesicles can function in this intermediate stage saturated solution. They can maintain encapsulation. So I'm moving to my second question now. How do these membrane compartments fare after complete dehydration? What I've seen is that dehydration disrupts encapsulation by fatty acid vesicles, but surprisingly, phospholipid vesicles are more resistant. So here in this top row, I've again prepared fatty acid vesicles, in this case with additional decanol and glycerol monodecanoate. I've prepared the vesicles with an aqueous fluorescent dye inside of them. And then again, I'll add a hydrophobic dye right before I take the images. And so to this initial population of vesicles, I dehydrate, I give it about 30 minutes of complete dehydration and then rehydrate. And I see that the vesicle itself does reform, but there's no longer a high concentration of fluorescent dye inside the vesicle. So what I think has happened is that all the dye that's initially encapsulated upon dehydration and rehydration gets released to the surrounding solution and mixed. And so now we're just looking at a uniform image here in the aqueous dye channel. Now, in contrast, if I do a similar experiment with phospholipid vesicles that I've prepared in an analogous way with fluorescent dye inside of them, dehydrate, rehydrate, vesicle reforms. But here in this case, there actually still is a high local concentration of the fluorescent dye inside the vesicle. So the encapsulation by phospholipid vesicles has been maintained. Okay, so that gives us now some understanding of what happens to vesicles when you dehydrate them. Prebiotic fatty acid vesicles will rupture and release their contents into the surrounding solution when it rehydrates, but phospholipid vesicles actually can maintain that encapsulation. And so we speculate, maybe this could have been a selective advantage for protocells developing in these environments to begin incorporating phospholipids to kind of maintain their individualization during these dry periods. Okay, so now I'm going to move on to my third question, which is, are membrane compartments compatible with biopolymer formation during dehydration? So we know that dehydration makes condensation reactions more favorable. A good way to see this is to consider the reaction of two amino acids to form a dipeptide. So here, these two amino acids are serine. They react to give a dipeptide, which is just two amino acids linked together by a peptide bond. And if you think about a protein as a chain of amino acids, this dipeptide is basically the first step towards making a protein. So again, two amino acids react to form a peptide, and water is also a byproduct of this reaction. So if you imagine dehydrating your sample and removing all the water, your chemical intuition might suggest that that's just going to shift the equilibrium of this reaction to the right, and you'll form more dipeptide product. Now, in practice, what's been seen is that unmodified amino acids actually require very high temperatures during dehydration, at least 90 degrees C, in order to generate peptides. So I've been working with this molecule called serine ethyl ester, which condenses into dipeptides at much lower temperatures. So the experiments I'm going to tell you about were done at 55 degrees C. Now, serine ethyl ester is basically, it's the same amino acid serine, but I've added this 2-carbon ethyl ester tail to the C-terminus. And so this reacts to give you similar dipeptides with the similar C-terminal ester. Now, you might say to me, okay, this makes your experiments easier, but is this really like a good prebiotically motivated choice? And I would actually say yes, this is a good model for prebiotic syntheses and also for peptide formation in modern biology. So as a model for prebiotic systems, I'm thinking about these prebiotic depsipeptides, which form from a mixture of hydroxy acids and amino acids upon dehydration. And it's a polymer that has a mixture of peptide and ester bonds, and the esters play a similar role there. And in modern life, this molecule here is called an aminoacyl tRNA, and it's just an amino acid with a C-terminal ester to the final nucleotide of a tRNA. And so that is, you can think about that kind of similarly to serine ethyl ester here. So I would say, yes, it is a good model system. Now, my big question is, do membranes affect condensation reactions during dehydration? And this has been studied in the literature to some extent. In this pair of papers, these authors have mixed nucleotides with phospholipid vesicles and dehydrated them. And they do see the generation of polynucleotides. And they also measured, they showed that the nucleotides actually became arranged within these stacks of lipid bilayer in the dehydrated sample. Some more recent work has mixed the amino acid glycine with phospholipid vesicles, and they actually report that the yield of peptides is a lot lower in the presence of membranes compared to the absence of membranes. So I want to come back to these questions, but to focus on prebiotic membranes and activated amino acids. What I found is that dipeptides form at least as fast in the presence of membranes compared to the absence of membranes. So again, here I'm starting with serine ethyl ester. I dehydrate it to get linear dipeptides and cyclic dipeptides as well. One limitation of these experiments is that I actually can't distinguish between those two. But here I'm plotting the total dipeptide percent yield as a function of time after complete dehydration. So what that means is that at t = 0, the sample is completely dehydrated, and I'll let it incubate at 55 degrees C for some number of hours before I rehydrate. Now I've got this red and the blue line on this plot, which corresponds to membrane and no membrane. So in the membrane case, I've just got my standard decanoic acid vesicles in solution. And in the no membrane case, the initial solution contains heptanoic acid, which is actually not, it's a similar fatty acid, but it's not hydrophobic enough to assemble into vesicles in solution. So it's a good control for not having membranes. Now, what I see from these experiments is that the initial rate of peptide formation is actually slightly faster in the presence of membranes compared to the absence. And we've kind of taken a conservative approach to talking about this data, and we just say that dipeptides form at least as fast in the presence of membranes than in the absence. Now, you might say to me, what about longer oligomers? And I did measure that as well with tripeptides. And in my system, what I've seen is that the yield of tripeptide is actually much lower than the yield of dipeptide. So it's only about 1% yield of tripeptide, although it seems to be a similar trend where the initial rate seems to be at least as fast in the presence of membranes. Now, I was still wondering what is actually happening to the membrane itself during dehydration. And to study this, I worked with a collaborator doing atomic force microscopy. So this is a technique that's used to measure the detailed topography of a dried surface. And basically what we do is we drag this AFM cantilever along this dehydrated sample, and we can detect very small changes in the height of the sample. And we can use this to construct images. So my finding here is that dehydrated membranes do form bilayer sheets, which is consistent with what some past literature has shown. So here in this image, the color corresponds to the height of the sample. So in this first column, we dehydrated decanoic acid vesicles, and we saw a mixture of these regions of these kind of flat regions and a mixture of these ribbon regions. To understand what this was, we compared to some control experiments. So when I dehydrated phospholipid vesicles, which we expect from previous studies to give us sheets of stacks of bilayer, we saw these similar flat regions. And so we thought, okay, the flat regions in the decanoic acid, the dehydrated decanoic acid sample must also be sheets of bilayer. I did a similar negative control with heptanoic acid micelles. So these don't form membranes, they just are micelles. I dehydrated them, and we saw these ribbons. And that matched, that helped confirm our picture of the ribbons from the decanoic acid case. It makes sense because decanoic acid vesicles are always in equilibrium with micelles, and so we have a mixture of these sheets and ribbons. Now, the final thing I did here to give us more confidence that we were looking at sheets of membrane was to draw these white lines through the sample and to measure the height as a distance along that white line. And we saw that that height changes in discrete steps in both cases, in the decanoic acid and the dehydrated phospholipids. So that just gave us more confidence that we're looking at these sheets of bilayer. And all of these data are consistent with this kind of schematic that I've drawn here for peptide formation occurring within dehydrated sheets of these prebiotic membranes. Okay, so to sum up, the behavior of vesicles that I've seen in these model evaporative environments is consistent with what we think was important for protocell development. So I'm just going to restate my key findings here. Encapsulation by fatty acid vesicles survives flocculation in saturated salt solutions. Encapsulation by fatty acid vesicles does not survive complete dehydration, but encapsulation by phospholipid vesicles does. And number 3, fatty acid vesicles allow peptide formation during dehydration. Okay, so I just want to thank the organizers again for giving me this opportunity. And thank you guys all for listening. And then just take a second here to explicitly thank all my advisors and collaborators and funding sources that have given me the opportunity to do this research, and all my friends in the Keller Lab and the astrobiology program who've been really supportive and made it fun to do this work. So with that, I'd love to take any questions if there are. Thank you.
All right. Thank you very much, Zach, for that very fantastic talk. So we have some time for questions, and what we'll do now is open up the chat. And so feel free to either just type in a question, or you can raise your hand and we'll unmute you as well. And so I'm sure there's plenty of questions in the chat. Oh, here's one. I don't know if you can see that, but I'll read it anyway. This is a question for Roy. Have you compared behavior of different RNA vesicle systems depending on the composition of vesicles? Could you please comment on this if you did? Thank you.
Yeah, we haven't, but actually years ago, Irene Chen with Jack Dostak showed interestingly that vesicles with RNAs will grow at the expense of vesicles without RNAs. And the idea was that it was an osmotic effect exerting tension on the membrane. of the vesicles with the RNAs. And so they were able to take up free fatty acids from the medium. But no, we haven't ourselves.
And there's another follow-up question for Zach. Maybe I missed it. How did you prepare vesicles? Have you used some of the guided assembly procedures or something else?
Yeah, good question. So for fatty acid vesicles, basically I'll make a solution of my cells. So deprotonate all you take a solid sample of the fatty acid and deprotonate it and make my cells, then lower the pH. And if I want to include a fluorescent dye inside the vesicles, I have to mix that in while I'm initially preparing them, and then remove the dye from the outside using size exclusion chromatography.
All right. So Burkhard Seelig has a question. Please go ahead and ask.
Yeah, hello. Thanks for the very nice presentation here. My question is to Zach. Did you look for peptides that are longer than tripeptides? I know the yield probably goes down, and at some point you hit your detection limit. But of course, everybody probably agrees that longer peptides would be also interesting.
Good question. No, I did not look for those. The reason is our technique, we were doing mass spec and we were comparing to an isotope labeled internal standard. And so it was actually kind of hard to procure those. So I didn't have an internal standard for anything longer than 3. But yeah, that would be interesting to do if we could have gotten those.
How did you determine the yield? So you talked about a % yield or so with mass spec. That's probably difficult or not possible.
Yeah, so we were doing, to do quantitative mass spec, we were comparing the signal that we got from our compound, this dipeptide, to an isotope labeled internal standard.
Yes, that
and
makes sense.
yeah. And so we did that with serine, with the monomer. I mean, I've actually got a step where I'll convert it. I've talked about this ethyl ester, but we'll convert it into a free C terminus. And so did that with serine, dipeptide, and the tripeptide.
Thank you.
Yep.
All right. There is a lot of questions. I'll try to go through as many as I can. Let's do one for Zoe. Let's see. So Mohit asks, as we have been told in our schools that life evolved with simple compounds and inorganic compounds, is it really possible to use the same and make life in lab? Oh, that's a good question.
I think that's a great question. And I would say we're trying very hard. And this is not just me trying very hard. I would say one of the labs that I spent my PhD in, like Jack Shostak's lab, that's trying to get to, can you make life in the lab? So we're trying, because if you can make life in the lab, you can learn a lot more about how life might have started on early Earth. So I would say it's an open question. I certainly hope so. Maybe that will come even before we get the first, tentative or definitive detection of life on another planet, but we'll see how it goes. Thanks.
All right, very, very good answer. So I think Helen Greenwood had her hand up next. If you'd like to go ahead and ask a question. Oh, can you unmute? Oh, maybe some technical difficulties. Oh, there we go. Yeah.
Membraneless organelles or coacervates are another type of protocell. And they can also work with lipid membranes. So I wondered if you had any comments on those.
Sure, I could answer that question. That's a great question. There was a paper that came out about that relatively recently. I think that the saturated salt would be a problem for those coacervates, but what they showed in this paper was that upon dehydration and rehydration, some, like for example RNA, I think actually at equilibrium will partition into those droplets, and so that in that sense is more attractive than a vesicle, where a vesicle, once it exits the vesicle, it's not going to spontaneously enter again in a high concentration.
Thank you.
I guess I'll just add that, the coactivate work has really been fascinating over the last few years. I mean, historically, that was the original idea. I think if you go back to the 20s,
That's coming
1920s.
back. I have a chapter coming out on it, a membraneless organelle lipid phase separation book with a chapter on lipid phase separation at the origin of life. So that'll be out.
I was just going to say, but what appeals to me about the fatty acid view is that there is a smooth path to phospholipids, actually, because Zach didn't emphasize that, but the two hydrophobic tails on a phospholipid are, of course, fatty acids. So there's more of a straight evolutionary line in that respect.
Zoe, I don't know if you have thoughts.
I'm gonna leave it to, I think, Roy and Zach. Their answers were very, very wonderful. I don't think I can contribute much.
Thank you very much.
All right, thank you very much. A question from Dave Deemer. Would you like to go ahead and ask?
Yes, hi, everybody. And Zoe and Zach, those are very nice talks. Thank you. I got a comment and a question for both of you. I'll start with Zoe. You use the entire ocean as a constraint on how much cyanide might have accumulated. That's a wise choice because the ocean hasn't varied that much over the past, say, 4 billion years in terms of total volume, we imagine. But when you think about the amount of ferrous iron in the ocean, there is a way to kind of get at least an order of magnitude, maybe a more accurate idea of how much iron might have been in the prebiotic ocean. We have a pretty good idea about how much iron ore there is on the Earth today. The iron ore is produced by oxygenation of ferrous iron. It precipitates basically as rust. And it's very impressive to go to Australia and see iron mines many hundreds of meters deep as they ship all this iron ore off to China. But instead of shipping it to China, let's dissolve it in the known volume, the estimated volume of the ocean. I've done that calculation and it's kind of surprising. It comes to about 2 micromolar iron in the ocean before all that rust precipitated out. Now, is that enough iron to interest you with respect to making ferrous cyanide and so forth?
That's a wonderful question and a fascinating backstory there. So I think you bring up kind of the big main question that still lingers in my mind, which is what concentrations do we need for prebiotic chemistry? And that's not been explored down to the lower limits because chemists have a hard enough time just getting their reactions to work under the most ideal conditions in the lab. So I think, exploring the lower limits for productive chemistry is definitely something. I've kind of had in my head the range of tens of micromolar and up might be plausible, but that's very much kind of by a back of the envelope kind of estimation, gut intuition feeling. I personally have gone down to like 62 micromolar of a copper cyanide and 125 micromolar cyanide and gotten results that work out of that. So is 2 micromolar enough? Maybe, maybe not. It might depend on what else is going on in your system, but I think that would be fascinating to try to quantify further. So
yeah, I agree. It'd be a very nice thing to study.
Yes.
For Zach, when we began to first make membranes from the Murchison meteorite back in the 1980s, we saw that they were highly fluorescent. So we would isolate the fatty acid content of the meteorite by two-dimensional thin layer chromatography, and they were in a fluorescent spot. So the point is that some polycyclic aromatic hydrocarbons come along with those membranes. With Matt Groen in Pascal Ehrenfreund's lab, we began to put PAH into fatty acid membranes. It had a stabilizing influence, something like cholesterol does today. So I think there is another interesting additive that we can play with as we begin to think about stabilizing fatty acid membranes along with, of course, the fatty alcohols and monoglycerides that you use for the decanoic acid. So I thought it was very interesting. And when you put a polycyclic aromatic into a lipid bilayer, it becomes a pigment. It can pick up near blue UV light, and it can do photochemistry. And to my knowledge, nobody has really ever followed up on the idea that polycyclic aromatics might have been one of the first photosynthetic pigments that just come along with the self-assembly of membranes.
That's a fascinating point. I would love to, thank you for bringing that up. I think complexifying these membranes and these systems, could just lead to really, really interesting behavior.
Okay, that paper was published. You can find it just a couple of years ago. Groen is Groen, and Pascal has her name on the paper as well.
Okay, great, thank you.
All right.
And Zoe is thinking about including those, Dave, in terms of a protective effect for things inside the vesicle. So, yeah, definitely an important factor to consider.
All right, thank you. I think Sukrit Ranjan, you had your hand up next.
Hi, thank you both for really wonderful talks. Zoe, I had a question for you regarding the loss of ferrocyanide due to the photoaquation process. Have you considered the potential role of the back reaction in your analysis?
Yes, so we were monitoring the total loss of ferrocyanide, not the production of the photoaquated species, because the back reaction is occurring potentially very fast at certainly basic pHs. So that back reaction is occurring in what we're actually also measuring, and we're measuring the loss then of the photoaquated species and total loss of ferrocyanide from solution. So I think we take that into account, but there's definitely the back reaction occurring at different rates depending on the pH especially. So that it can get a little more complicated than probably I alluded to.
Do you find you tend to agree or disagree with the argument of Turner and Catling then? Because I think they come to different conclusions a little bit.
Yeah, so it would depend again on pH and on the conditions on earlier. So, and also how consistent the UV radiation is. So if you get to a dark period, then you probably can restore a lot of your photoaquated ferrocyanide back to ferrocyanide. But if you have this continuous radiation, then you have the potential for continued destruction and loss from solution.
Got it, thank you.
Yeah, thanks.
All right, thank you. We have a question from the chat from Christian Luca. This is for both speakers, he says. Very great presentations. When it comes to prebiotic chemistry research and origins of life, is the radiation level accounted for, which was different than it is now, of course, on the cosmological geological timescale of Earth's history? Could looking at the influence of cosmic rays help further origins of life research? As I have read that cosmic rays are instrumental in leading to the handedness, chirality, in simple organic molecules. Thank you. So I guess either Zoe or Roy or anybody can jump in.
Leave that to Zoe.
So I could jump in and say, yeah, I think the influence of cosmic rays, especially on chirality, has been suggested, and there are some really fascinating suggestions that could be leading to the influence of why life has the chirality that it does today. I am by no means an expert in that, but I think as prebiotic chemists and early Earth environment people, if we consider all of the resources available to us, including these cosmic rays, of course, those should be considered and see what interesting chemistry could result from there. So I think it's a great thing to explore.
All right. And I think in the interest of time, we can ask one more question. And I know Amaya Selva had her hand up, so you can go ahead and ask your question.
Hello, my name is Amaya and I'm from Sri Lanka. I'm still 14 years old. And my question is, what is the contribution of prebiotic chemistry towards the process of abiogenesis?
It's a very good question.
Yeah, that's an excellent question. Zach or Roy, do either of you want to jump in?
I was waiting for you.
I would just say I think that helps us understand the chemical inventory that would be available for the origin of life, for abiogenesis. Once we learn, what can actually form in the absence of life, prebiotic chemistry.
Yeah, no, it's a critical part of the story, and I think that's what Zoe really was focusing on in her first segment.
Yeah, I would second everything that Zach and Roy said. I would say, abiogenesis gives you the building blocks or the molecules that then at some point abiogenesis transitions into prebiotic chemistry, which then transitions into life. And there's probably not a defined concrete, cutting off line to get from one stage to the next, but it all merges together, in my opinion.
Great question. Thank you for the question.
Yeah, and it's awesome that you're 14 and you're here and asking wonderful questions. Thank you so much.
Yeah, that's fantastic. That's amazing. Thank you very much. All right. So I apologize if we didn't get to your question. There's always another chance, and I'm sure our speakers wouldn't mind if you just emailed them directly with your questions. Is that all right with you guys?
Absolutely.
Yeah. All right. So with that said, thank you, everybody. And thanks again to our 3 speakers today. I'll go ahead and give them a clap. Thank you very much. And so just one last thing. If you are a young postdoc or graduate student and would like to talk, please get in touch with either me or the other seminar coordinators, and we'll be happy to schedule you in. So we'll see you next in 3 Thursdays. All right. Thanks again, everybody.
Thanks, Albert.
Yeah, thank you.
Thank you.